So if this is the eye and this is the cornea, then we will have the retina somewhat like this. And whenever we look at light, it will pass through the cornea and other structures, and it will hit a certain type of cell in the retina. And we have two types of photoreceptors in the retina, which are called cones and rods. So it looks somewhat like this, I guess. And this is a rod, and in these rods there are membranous discs, which will look somewhat like that. So this is a closer view. And inside of these discs there will be something called retinol. And whenever light hits this retinol, it will cause a conformation change in the molecule. And when retinol changes its conformation, it will become active. And when retinol is active, it can activate another molecule called opsin. And this opsin will also become active, and it will activate something in the membrane of this disc that is called transducin, which is another enzyme. And furthermore, this transducin will activate another molecule in the membrane, which is called phosphodiesterase. And the job of phosphodiesterase is to convert cyclic GMP to GMP. And that is important because cyclic GMP will have an effect on the cation channels in the membrane of this photoreceptor. And be aware this is on the outside of the cell. So cyclic outside of the membrane, So cyclic GMP will stimulate this channel to stay open. And this channel is a cation channel, as mentioned, which will cause calcium and sodium to pass through. And when that happens, this cell will become depolarized. It will become more positive. But given that it's cyclic GMP that does this job, then when light hits this membranous disc, it will convert more cyclic GMP to GMP. So we will have less stimulation of these channels, as mentioned. Therefore, less cations will leak into the photoreceptor. And when we have less cations in the inside of this photoreceptor, it will become less positive, meaning it will become more negative. And this is the nucleus, by the way. And at the end of the photoreceptor, at the nerve terminal, here are other channels that are voltage gated, meaning they will open as a response to a depolarization of the cell. So if this cell would become depolarized, this channel would open, and this is a calcium channel. And we have more calcium on the outside of the cell than the inside. So calcium would have gone into the cell like that. But light causes the photoreceptor to become more negative. So calcium won't enter the cell. And this calcium will also not trigger the release of neurotransmitters, which it normally would do. And these are vesicles containing glutamate, which is the neurotransmitter, which would normally fuse with the membrane and release glutamate into the synaptic cleft. And this photoreceptor is in contact with another cell, which is called a bipolar cell. But this glutamate has a special effect on this bipolar cell, that it will actually inhibit the cell by causing cations to leave the cell when stimulated. So, to sum up, when light hits this photoreceptor, less glutamate will be released in this synaptic cleft with the bipolar cell, and that causes less inhibition of the bipolar cell. And when we have less inhibition of that cell, it will in effect become stimulated. So when it becomes stimulated, it will become depolarized, and this positive signal will go through the cell as an excitatory postsynaptic potential, and that will trigger the opening of calcium channels at the nerve terminal. So calcium will flow through, and we also have these vesicles containing glutamate, and the calcium will help the process of exocytosis of these vesicles in the nerve terminal. So glutamate will be released into the synaptic cleft, as mentioned before. And this bipolar cell is in contact with one last cell that is called the ganglion cell. And these ganglion cells are known for having really long axons in relation to other cells. And there will be a bunch of them. So whenever glutamate is released in this synaptic cleft, this ganglion cell will become stimulated, in the sense that it will become depolarized, and it will trigger an action potential, unlike the two other cells mentioned before. They will only generate EPSP. And this positive signal will go through the optic disc of the eye. So if this is the eye once again, all the ganglion cells, which is marked in red, will go through the optic disc and gather there together, and they will go out as the nervus opticus, which is also called the first, the second cranial nerve. And it will send this visual information to the visual cortex in the occipital lobe.